Alright guys, this is item number 22 in your Kingdom Animalia table of contents. This is called the Population Genetics and Evolution Lab. I expect you to read and highlight it, and it is a doozy. So this lab's going to take us several days to do. Again, it's about Hardy-Weinberg. You've got to know the formula. I got to know what circumstances you can use it with. And we're going to actually study a couple of things here. So under Activity 1, you're going to need to go over to my cabinets by microscopes. There's a drawer that's labeled, I think, pH paper, but you're actually looking for the PTC paper. You guys probably tried this in biology last year, the year before, but if you are a taster, it's going to taste sour, it's going to taste bitter. If you're a non-taster, it's going to taste like paper. So there's two different types of containers. You want a control and you want the PTC paper. Try the control first. Make sure no gum, no candy. You might want to grab a cup out of the household cabinet, take a sip of water in between. So you're going to record that data actually on the data chart labeled Genetics of a PTC worksheet. So there's going to be a place for you to put percentage of our population, so out of 100%, how many of you are tasters, and percentage of the class that is a non-taster. And then they actually give you data for actual North America, okay? And then you're going to compare p squared plus 2pq to q squared, which would be your homozygous recessive, our non-tasters. So you're going to compare our allele frequency with the allele frequency of North America and see if they coincide. And then you're going to answer questions 1 through 3. Make sure that those are in sentence format. So I encourage you to read Activity 1, do procedures 1 through 9, and fill out the correct chart. You are then going to be working on activity number 2. For the rest of these activities, you're going to use this stack of cards. There are capitals, for our dominant, and then there are, somewhere in this stack, lowercase for our recessive. These cards are in the cabinet that say AP Biology. You're also going to need some sort of coin. So I'm sure somebody in the class has enough of those. For activity 2, there's actually case 1, case 2, case 3, case 4. You could probably do the PTC test and maybe case 1 in one day, but that might be cutting it. You're probably only going to get the other one cases, case 2, case 3, case 4. It's going to take you several days to do this lab, because I would hate for you to maybe do one case, start the next, and then completely run out of time and have to start that case back over again. So case 1 is like the ideal situation, the ideal Hardy-Weinberg population problem. You have got to read the questions or the directions very, very carefully, okay? You've got to know what P is asking for, P squared, Q, Q squared, 2PQ, and so on. Remember, 2PQ is heterozygous. You've got to read these directions, and then your data for that one goes on the Hardy-Weinberg equilibrium worksheet. There is basically a worksheet for each case, so that's on the first one. Then you go and do case 2. That goes on the one that says selection. And then you go on to the one that says case 3. That is the one labeled heterozygous advantage. And then finally, case 4, genetic drift. This lab is a lot of recording of data and then data analysis, and then some of the cases also have questions with them. There is no hypothesis, therefore there is no rerun. But keep in mind that you are going to have to take several days to do this, and be very cautious about doing more than one case in a day and running out of time, because some of these have multiple generations. In years past, the class has been small that we've had to repeat case 1 more than one time, repeat case 2 more than one time, in order to get enough data and to make sure our data isn't by chance, but it's actually the real deal. Again, that is item number 22.